And let's turn to Dr. Barbara Rehermann. She's the chief of the Immunology section, Liver Disease Branch at NIDDK and NIH, and she'll be speaking on immunomodulatory strategies. Barbara?
Thank you. Can you see the slides?
Yes.
Yeah.
Yes. So I would like to thank John and Rajan Koshi for the invitation to speak at this meeting. And like Stefan and John, also congratulate Rajan on his recent retirement and thank him for all of the decades of work and his help to all of the HBV researchers worldwide. His work started at the Max Planck Institute in Munich in Germany and then went over decades at NIH. And I know you're in the audience, Rajan, and I hope you will join the discussion. So I was asked to review immune modulatory strategies. So these are my disclosures. So I'd like to start by asking why cure of chronic hepatitis B is so difficult from an immunological perspective. And it's important to note that the differential disease presentation and outcome really results from differential host-virus interaction due to the different age of infection. So we know, and it's very well established, that HBV infection during adulthood results in an acute, often severe phase of liver inflammation that is attributed to a strong adaptive immune response. The disease is self-limited in almost 95% of cases and results in lifelong cellular and humoral immunity that is protective but not sterilizing because cccDNA persists. So this successful natural immune response is well characterized, biomarkers of protective immunity have been established, and prophylactic vaccines are widely available and effective. In contrast, however, most of the 254 million infected persons worldwide and the many more who are yet undiagnosed have been infected at a completely different age, perinatally or as young children. And there are two important factors that I would like to emphasize. So despite exceedingly high HBV levels in the early years and decades after infection, this does not result in any clinical symptoms. And we do not understand why this virus, while inducing an immune response at that stage, does not really result in any inflammation. And secondly, death due inflammation due to decompensated liver cirrhosis or cancer typically occurs much later than after the reproductive age. And this points to a co-evolution of the virus and the host. And indeed, HBV sequences have been found in 400-year-old preserved mummies, in 3,000 to 7,000-year-old fossils from the Bronze and Neolithic ages. And the entire family of HepDNA viridae is thought to have diverged 400 million years ago from non-enveloped DNA viruses in fish. So overall, this shows that there's a long process of co-adaptation and co-evolution of HBV with us, humans, the current hosts. And this is the reason why chronic hepatitis exists and is so difficult to eliminate. So we are simply many, many years behind of the virus. The virus has studied the immune response much earlier than we have. And it's questionable whether we can revert this, but definitely we may be using HBV also as a tool to learn about host immune responses. So it's well known that there are these different phases of chronic HBV infection. As I mentioned initially, there's exceedingly high HBV DNA level and no liver inflammation detectable, no ALT inflammation, and also very high HBs antigen levels. And it's not really clear in the natural history of the disease why this converts to an HBe antigen positive chronic hepatitis phase with changes in HBV DNA levels and inflammation. I think much more immune research on the natural history of HBV, of chronic HBV, is still warranted. What do we aim for? We are aiming for functional cure of chronic hepatitis. This means that HBV DNA serum levels have to be below the level of quantitation, and HBs antigen should be undetectable. Anti-HBs can be present or not present. The loss of HBs antigen alone is associated with beneficial clinical outcome. So these were the criteria established for functional cure at the 2016 AASLD and EASL Treatment Endpoint Conference. More recently, at the 2022 conference, partial cure has been advocated as a next step, as a first intermediate step. And this is the same. Serum HBV DNA have to be below levels of quantification, and that has to persist a minimum of 24 weeks after end of treatment, and after that long term. And HBs antigen levels, if not undetectable for partial cure, can be below the level of 100 international units per ml. The patient has to be HBe antigen negative. So this requires, as we have heard from previous virological talks, a control of HBV cccDNA, the transcriptional template for HBV, and that is established as a mini chromosome in infected patients. This can be achieved via turnover, dilution basically, among progeny of HBV cccDNA containing hepatocytes, but the half-life time of cccDNA is long, and the turnover of hepatocytes is small unless there is significant inflammation by immune responses. Cytopathic mechanisms, death of HBV cccDNA carrying hepatocytes, non-cytopathic mechanisms of degradation, cytokine-induced, and silencing inactivation of cccDNA are important. And as you can see, many of these factors depend on immune control. In addition to control of HBV cccDNA, we need control of integrated HBV DNA. Seminal studies by Bill Mason have shown that HBV DNA, also non-replicative, integrates throughout all of the chromosomes, except here the Y chromosome at the bottom. And this occurs very early after infection. It already occurs in the E antigen positive phase of viral infection, the first phase, when there's no sign of liver inflammation or clinical disease. Bill Mason has quantitated here the number of hepatocyte clones with integrated HBV DNA. You see that he found 15 to 39 clones in the HBe antigen positive stage in biosamples. And then this increases, and the number increases, and the clone size also increases significantly in the HBe antigen negative phase of infection. And this integrated HBV DNA is considered to be a major source of HBs antigen levels in the blood. So what are the immune modulatory strategies? The first strategy, of course, is to activate the innate immune response. It's well known that HBV does not induce interferon alpha and interferon stimulated genes, but it's a good sign that it is sensitive to them. There's an experiment of nature. We now know that HBV, HCV co-infected patients control HBV replication because HCV induces interferon and ISGs. And then if HCV is cleared with direct acting antivirals, this results in an HBV reactivation. So this endogenously produced type 1 interferon response due to HCV infection can control HBV. In vitro, all of these mechanisms here have been shown to be effective antiviral mechanisms, effective also against cccDNA, but this is in vitro and they depend on very high interferon levels. In patients, treatment with pegylated interferon alpha remains the backbone of many of the new immunomodulatory combination therapies. The effect is a very rapid induction of interferon-stimulated genes and also a very rapid activation of natural killer cells that are very responsive to interferon because they express high levels of the interferon alpha-beta receptor. And we have shown here in the study that this occurs in both liver and in blood within 6 hours after the first injection of PEG interferon alpha. And there can be a differential response in patients that we have shown to correlate with HBs antigen level decline. And so this very first response is important. And the greater the response, as shown by the patients in red, the better for HBs antigen decline later on through the study. And the patients who do not show an increase here in natural killer cell activation, the black dots, they do have a response, but it occurs later. And this depends on the pharmacokinetics of interferon levels in the blood. And it depends on pre-existing levels of antibodies against PEG, as we have shown here. So HBs antigen loss occurs in 2 to 3% of patients at the end of a 48-week of treatment regimen with pegylated interferon alpha, and then increases long-term after the end of treatment. But it's associated with significant interferon-induced side effects in the patients, and the response can increase if PEG interferon is added on to long-term nucleoside analog therapy. The patient is infected with HBV genotype A, which the majority of patients worldwide is not, unfortunately, and if the pre-treatment level of HBs antigen is low. So how can we otherwise activate innate immune response? A broader activation of innate inflammatory responses, including IL-6, TNF-alpha, and type 1 and type 2 interferon, has been tried with TLR7/8 agonists. Monotherapy with these agonists have minimal clinical efficacy, but I would like to mention one combination therapy study that was presented at the last AASLD meeting. So in patients that received nucleoside analogs, there was an addition of a direct-acting antiviral, an HBV siRNA that blocks S antigen production. And in addition, immune modulators, either a TLR7 agonist or PEG interferon alpha were added. And as shown here, this increased the percentage of patients that received S antigen loss at the end of treatment, here shown in the light orange, and at the long-term treatment endpoint, week 24, after end of treatment, shown in orange. However, as you can see, the end of treatment time, the week 24 follow-up treatment is about 13% of patients losing S antigen with this combination treatment with the TLR7 agonist. But if you, instead of the TLR7 agonist, include PEG interferon, the response is even greater, and it's 22%. And the response, again, depends on S antigen levels being low prior to treatment at baseline. So what about adaptive immune responses? So we know that in chronic infection, HBV-specific T cells have signatures of exhausted cells. We know this depends on high antigen load and, as shown by Frank Cesari in studies in chimpanzees, on insufficient CD4 T cell help. This sets off an exhaustion program that starts with induction and long-term induction of the activation marker PD-1, CTLA-4, TIM-3, LAG-3, and then impaired effector functions, resulting further down in impaired mitochondrial polarization and accumulation of ROS, and in metabolic and epigenetic changes, in the end, in loss of some of these cells. The same has been described in HBV-specific B cells that are also dysfunctional in chronic hepatitis B. These cells can now be studied with fluorescently marked BAIDs, fluorescently marked HBs and core antigen. And as you can see, as in many other chronic infections, as in HCV and HIV infection, there's an increase in atypical memory B cells in chronic hepatitis B, decrease in classical memory B cells, and there's reduced B cell receptor signaling, production of antiviral cytokines, plasma cell differentiation. So this, both at the T cell level and in the B cell level, has responded to blockade with of PD-1 in vitro and partial recovery of T cell and B cell function. However, in patients, this is quite a different story. We know that HBs antigen-specific T cell responses decline with age, and as you can see on the left, so the percentage of total HBV-specific T cells that respond to S antigen is much reduced once you're over the age of, let's say, 35. And this is specific for HBs antigen-specific T cells, where it's most prominent, as compared to HB core-specific T cells, where we see not such a correlation. So if we then try to block with PD-1 antibodies, this is going to be very difficult because we cannot completely reconstitute. If we try to replace HBV-specific T cells, for example, by bone marrow transplantation from donors who recovered from acute HBV infection into chronically infected recipients, this does work. So you see here, again, an experiment of nature. You see that this patient develops ALT levels increases 28 weeks after bone marrow transplantation, and then seroconverts first to anti-HBe and then to anti-HBs. This is associated with an increase in HB core-specific T cell responses. The inverse is also correct. If you transplant an HBV-infected liver into a patient who has anti-HBV immune responses, these immune responses also control HBV in the infected liver. So this is an example that supports the development of specialized, personalized treatment with specific T cells that express very specific HBV-specific T cell receptors or chimeric antigen receptors, as shown in this study here by Tan et al. In a pilot study. This can happen also spontaneously. So we have seen an increase in HBV-specific T cells in patients who seroconvert from HBe antigen positive to anti-HBV positive and HBe antigen negative. And this has also been associated with an increase in functionality of these cells. So here you see in light green graphs the number of HBV-specific T cells when you put them in an in vitro cell culture. And then when you stimulate them with the antigen, you see, as shown by the dark green bars, differential level of expansion of these T cells. So you see greater expansion in those patients who are HBe antigen negative. So this increased T cell number and increased functionality of T cells that we see in HBe antigen negative patients as compared to E antigen positive patients is not associated with increased liver disease. That is important because the liver enzyme levels are lower after seroconversion than in the E antigen positive stage. So this means
I'm sorry, Barbara, can you wrap up, please? Just
yes, I will. So this means
so much.
That an increase in T cell function is not always associated with increased disease inflammation, but rather with increased immune control. Checkpoint inhibitors I mentioned, anti-PD-1 alone resulted here in this study in S antigen loss in a single patient. Newer studies have combined it with seropolitic vaccination with chimpanzee adenovirus prime and MVA boost. And this results in decreases in HBs antigen levels, but the number of patients is still very low, and the HBs antigen levels prior to treatment are also low. Overall, as published by NIAID in this very interesting and great recent review article, HBs antigen loss in clinical trials is associated, the strategy is to combine different antiviral treatment and immunomodulatory regimens that always include nucleoside analog treatment as a backbone and also often include pegylated interferon alpha. Overall, to summarize, we can replace the inefficient T-cell response and antibody response, B-cell response in chronic hepatitis B as shown in the left. That is one strategy. There are newer strategies in mice, as shown here. We can induce bispecific antibodies, which would make this more as an off-the-shelf treatment, but only shown in mice, not yet in patients. Or we can try to engineer continuous production and better antibodies against anti-HBs antigen, engineer humoral immune responses. Restoration treatments, as shown on the previous slides, include therapeutic vaccination in combination with all of these other new treatment regimens, both antiviral and immune modulatory. From an immunological perspective, I'd like to emphasize that we also need to modulate the immune tolerant environment of the liver, and that any new experimental treatment, if we really want to understand what it does, we also need to monitor immune responses in the liver via fine needle aspirates. Thank you very much.